all of the experiments, all of the science is done to aid and facilitate decision-making through a process. And exploration, for the sake of exploration, usually doesn't happen quite the same way in industry as it does in academia. But we do have project types very similar to a typical transcriptome experiment, for example, where you have a group of samples where you're working to try and understand the relationship between the samples so you can have insight onto some process. And then lastly, a category which I think is, I wouldn't say it's unique to a situation like ours, but it's perhaps a little easier for us, is the multi-meta-analysis across many different experiments. Because all of the samples are coming into the same group, and because it's within a company setting, we can actually dictate some standards for metadata. And so we can start doing meta-analysis across experiments that is a lot harder in the public domain because you have such a diversity of different data types. And so these are the 4 types of projects that my group handles. And we use the standard types of genomic sequencing and RNA sequencing type approaches. One other tangent that I want to throw in here is that we're a global organization. We have sites all over the world. So our samples come in from many different places at many different times of year, and they often have biological deadlines. That means some samples have to get turned around very fast, and others can wait. And this is really just a reminder for me to make the point that within an industrial setting, there is that whole pragmatic layer has to be there for what science you do. Because science is there to help make a decision. There's no point, so you can have fast data, you can have cheap or expensive data, and you can have low quality or high quality data. It's actually hard to get all 3. It's hard to get fast, high quality, and cheap. And so you have to make decisions about what you want. And there's no point getting cheap data if it comes too late for the biological deadline that you have. If you miss a whole season in the field, then that's not so good. So you may want to pay more to get that data faster. At the same time, there's no point paying more to get data you don't need right now. So there's a lot of practical logistics involved in making decisions on how to use different types of assays within the company. So I talked about the types of projects, the types of information we're gathering. Pretty standard. There's functional information, and this is usually around trying to understand the process within the organism, the plant or the pest. The goal here is really to identify genetic elements that we believe are involved in that process so that we can potentially either target them with a new molecule or to interfere with that process through an RNAi approach, for example, or to even take a gene from that process and try and exploit that process to amplify the effect. And for that is transcriptomics, epigenomics, those kinds of things. Another class, because we are a breeding company, there's a lot of what we call structural genomics happening, and that is really research that's aimed at understanding where in the genome particular elements are. And so these are things such as genetic mapping, characterizing variation within populations, tracking inheritance of a segment. So it's very pragmatic and very practical. You just want to see where that segment is. And for this is various types of DNA resequencing, like the whole genome or reduced representation. A lot of structural variation type analysis as well. So where in the company, or what factors has genomics made the biggest impact on? And I sort of alluded to this one at the beginning, but just the actual presence of reference genomes just makes a whole world of difference. And you really notice that when you start work on a species that doesn't have it. I think we take for granted, particularly in the human domain, I think you take for granted now that there is a human genome, and you know all the genes. Well, think back to when we didn't have that. Well, that's the reality for many of the obscure pests and insects that we work on within the company. So getting to that point makes a huge impact. Variant discovery, I think, is probably the biggest single type of information that Genomics has contributed within our company is the ease with which you can go and get information that distinguishes two samples, whether it be two lines of the same species, whether it be two cultures of the same bacteria that has mutated, but just the ease with which you can find the differences between the two things. And the fact that it has the precision and the ability to scale, it makes it light years better than hybridization for most things. And that sale within the company of shifting from hybridization-based assays to sequencing-based assays has made a huge difference. And then lastly, this one probably isn't so obvious, but it's just the ease with which you can do the experiments. The low cost and the quick turnaround has really lowered the bar for people asking questions. And that's a good thing. That increases the pace of discovery. It increases curiosity. So it used to be that people would say, oh, wouldn't it be great to do such and such an experiment? Oh, but I have to write a grant, and it will be 2 years before I get enough money to do it. Well, now you can do it in a few days. And we've noticed this within the company, that just having access to these technologies lowers that barrier. Makes people ask more questions, and they're more curious, and they're more able to iterate on those questions, which is a really good thing. So these are where it's made the biggest impact so far. And I think, at least from my perspective, the area of biggest impact in the future is plant breeding. So plant breeding at its heart is a cycle. So you start with some material, you introduce some variation by either doing new crossings or perhaps even mutagenesis. And then you grow them all out and you make some selections based on their phenotype. And then you do the cycle again. That's basic plant breeding. But basic plant breeding has a black box. On the left, it's completely unknown variation. So you don't know on what are you selecting. And your only ability to select is based on phenotype. And phenotype is slow, it's expensive, and it's very prone to environmental interference. So you can't guarantee that you're going to get the same answer 2 years in a row, for example. You throw in genomics onto this, you have the ability to completely, fully characterize all of the variation between any two plants at a scale that's never been possible before. And then, with enough of those data points on the phenotype, you can look for correlations and then make predictions about what the phenotype is going to be. And therefore, you can accelerate the whole cycle because you don't need to spend the time doing the phenotyping. So that's the whole basis of genomic selection, which has been incredibly powerful in animal breeding. And I think this is the future direction of all plant breeding, is that accelerating that cycle as fast as you can. Because the way you get to 300 bushels is to iterate many, many times in the shortest space of time. This is a quote from a very nice review, if you're interested in this topic, very nice review, just looking at the impact of these kind of technologies in the future of plant breeding. But it's not all rosy, right? So these are some of the great opportunities, but there are some real challenges. And certainly, this is from my perspective inside the company setting. One of the biggest challenges is infrastructure. And I think anybody who tells you they have infrastructure for genomics under control, I think either is not telling you the whole truth, or they haven't thought about it enough. Infrastructure is a challenge because the way biologists are using the data, and the way the biologists are generating the data is changing way faster than the IT people. IT people are changing their hardware. And the purchase cycle for big hardware is much longer than the purchase cycle for new sequences. And that is creating a big challenge. Add on top of that, from the hardware side, let's say you can solve that, you still have data management. And I think data management is a headache for the small lab. It's a bigger headache for the medium-sized lab. And it's a migraine for a company. I mean, it is a nightmare. And so getting a handle on data management is absolutely critical to do anything in this space if you want to manage the assets of a large organization when it comes to data. people are a big constraint, I would say. The number of people is one issue, so the number of people that are trained in these fields. So I have two phrases: digital biologists, so these are first and foremost biologists, but they have the skills to work with perhaps in the Unix command line. They have the skills to think about things in terms of big data sets. And then there's the bio-savvy computer scientists who are needed to tweak algorithms and come up with new algorithms to solve problems. But if they're not bio-savvy, they spend their time focusing on the wrong aspect, like make it fast. Well, that's great, but it doesn't need to be fast, it just needs to be accurate. So having the bio-savvy piece on the computer scientists is absolutely critical as well. And I don't think there's enough of either of these two populations to really meet the demand, which is really nice if you're one of them, because it means job security. But it's not great if you're a consumer of these skills, there's just not enough out there. And so I think organizations like Genome BC and UBC, the more you can train these people with these skills, the more you can fill these openings out in the workforce. And then the last constraint or challenge I see, and it's a pet peeve of mine, is that 95% of technology development in this field focuses on medical applications. And human-centric thinking goes into all aspects of that development. So there are a couple of things, a couple of points on this. One is just, if you go online, there's a lot of cloud-based sequence analysis platforms right now. Where you try getting a maize reference genome into them, you can't do it. Everything's on H37 or whichever the human genome reference is. And everything's marketed towards people working on things. So the sphere of things that are accessible for ag applications is a lot smaller. But the other aspect is, I don't think product development is driving to areas where ag can benefit. So what I'm trying to say is that it's okay to charge $1,000 for a human genome because consumers have credit cards, they have insurance companies, they have government health care. Corn plants don't have credit cards. So we have 1 million plots, and we would love to genotype high-density information about every plant out there in the field every summer. But there's no technology, there's no drive to produce a full genome characterization that costs 10 cents. Because why would you need 10 cents when you can get $100 for it, right? So I think one of the challenges working in ag is there's not enough motivation for the technology development companies to drive price down to make it fully applicable to ag because of the volumes we have to do. So I think that's about all I have. I just want to wrap up and say that from my personal perspective, genome technology, genome information is an absolute bedrock of future crop improvement efforts. And this is true for within a company that's selling big commercial crops, but it's also true across the developing world for improving crops that are more targeted towards their countries. I do a lot of work on the side with some crops for Africa, and genomic technology is helping them leapfrog whole generations of corn research that use much slower, cheaper, less informative technologies. Crops that have never had that research done are now leapfrogging because of genomics. So it's going to empower all crop improvement, and that's what we need to basically feed